It's nice to see so many of you who've been here many times before. How many were here 10 years ago? Significant number then. How many have been here? How many have never been here before? Wow, that's the biggest number of new people that I've ever had, I think, and I thought last time was a lot.
Don't forget 30 years.
Okay, who all was here 30 years ago? This is a wonderful place to be able to gather, and one thing that has made it so wonderful is simply the hospitality of the school and the basic nature of the school. And I came here in 1972, the second year of the school. And people often ask me how I wound up being at Evergreen, and enough have asked that I'm going to answer it, and then I'll and just a couple of other things. My kids had been born when I was in graduate school at the University of Rochester, and then we wound up going to the University of Virginia. For the first year, my husband insisted I sort of take the year off, and then towards the end of that time, I came to realize there were no women on the faculty at all in either biology or biochemistry there. Half the starting graduate students were women, and they were given the teaching assistantships, and the guys were given the research assistantships, and they weren't finishing. But still, I got invited to work half-time in the lab of a fellow named Rolf Benzinger, who had just been working with the people who developed the whole idea of recombinant DNA. And he pushed me then to apply for a grant, and I got it, and then for a second one 2 years later. And then I went to talk to the chairman about applying for one of 4 new positions the school had gotten. And he got absolutely irate that a woman with young children would have the audacity to consider applying for a faculty position, and said if I did, not only was I guaranteed not to get it, but he would no longer sign for my NIH 3-year grant. That was the biggest favor that anybody has ever done for me, even though it didn't seem quite like that at the time, because that was extreme enough to make my husband leave his very good tenure-track position in the astronomy department with a telescope right there, and he was an astrophysicist. And my parents had been sending us information about Evergreen. That was the first year of Evergreen. So we decided, and we'd been interested. So the first thing we did was we both applied to Evergreen. Within a month, we each got an invitation to have an interview, and those were carried out on the East Coast, sitting on a park bench in front of the White House. And 2 months later, we were offered jobs here. And this has been the absolute perfect place to be here at Evergreen in a lot of its ways of looking at the world. There were many people in the research community who really helped me to get going, and the person at NSF believed in what we were doing, and I had grants for every year up till 2000. From 1975 to 79, I was on the National Recombinant DNA Advisory Committee, as coming from a school where teaching the ethics and values of science was part of the teaching of science. And when I retired a few years ago, Evergreen was willing to let me keep my shared office with the students and keep access to being able to work on grants. It's only if I have students, I have to have somebody else sign for them. Since they're not paying me, I can't formally be in charge. Well, those people who are in my lab right now, stand up for a minute. Those are the people who've been, go ahead. You too. Those are the people who've been involved a lot in helping at this point make this possible. So, and with the people who are on the planning committee, please stand up for a minute. So there are people from all over. Some of you are missing still. There are a group of about 17 people from all over, yeah, you're all the way around through here, who've been the people who've done a lot of the putting it together and planning and inviting people. Where's Susan? Get up. She's the one who did the abstract book, and does it in such a useful way, and she deserves a very special thanks, as do Daniel and Matthew, who handled trying to keep track of all these amazing new people who were coming in and where plans kept changing. The person I'd next like to introduce this is the first time that a president of Evergreen has come, though a couple times before I've invited, and I would like to introduce our new president since the last meeting, George Bridges.
Thank you, everyone. Can you hear me in back? Is that good sound? What a great show of people here. My goodness, I just learned recently that we've got 250 people from 70 different countries represented, all interested in this fascinating subject of phage. Now, I will tell you that my training is in the social sciences, and so this was a steep learning curve for me. But what I've learned is, first, we have a gem of a faculty member in Betty Cutter, and to be frank, even though we think of ourselves, or as we think of ourselves here at Evergreen as a liberal arts college, our science faculty are among the most competent, established, and inspiring teachers that we have on our campus, whether it's in the life sciences and the earth sciences, in a variety of fields. So we're very proud of our science programs, the instruction they provide, and the working relationships they have with students. One of the beauties of Evergreen, and I will tell you that I am new, I'm just starting my second year. What a fascinating school it is, and what an inspiring group of students we have in their relationship with faculty. And one of the reasons that Evergreen drew me here, I was a president at another college, was the commitment to taking academically inspired knowledge and applying it to real-world problems, taking theory and putting it into practice. And if there's anything about phage that I do understand, or bacteriophage, is so much of the science of this is applicable to very important, critical real-world problems, whether they involve food safety or drug-resistant strains of bacterial strains. And the fact that this is such an impressive international gathering focusing on those issues and many others is just a joy to have you here. I will tell you that being from the Northwest, that the weather in Seattle area and Olympia is always this nice. Not true. But it is a beautiful time of year to have a conference. I hope you enjoy your stay. I hope you take advantage of the campus. It's in 1,000 acres of pristine woods and forest, as well as half a mile of pristine beach on Puget Sound. So walk around, enjoy the campus, get to know the college, and we're so delighted to have you here. And I look forward to hearing the presentations, some of the presentations this afternoon. I understand you had a great barbecue. And I have envy because I haven't eaten today yet, so I hope that the sessions are as exciting and as personally enjoyable to you as they are intellectually edifying. So thank you for coming. We're delighted to host this conference, and delighted, of course, that Betty's in charge.
Yes. Okay. One of the things that's made this particular year extra special for me is the fact that we have a special delegation here from various countries in Africa. And Toby, where are you, Toby? Yeah, stand up. Toby is basically responsible, and this is the sort of thing that can come up as a result of things that happened one year at the conference. She gave a talk about the dream that she had of establishing a nonprofit company that would really help bring phage for food safety over a wide swath of Africa. And she was so inspiring that when I was visiting my granddaughter in Tanzania for a month last summer and had a little time before the meeting in Liverpool of the Viruses of Microbes program, I decided to write to the people in Uganda and see if they'd like me to drop by for a visit. This was arranged on about 1 week. It turned out that they were putting on a Africa One Health program there. There were hundreds of people from various places, and they found a slot for me to speak then. But the one that pleased me more was that about 2 hours after I walked into her office, Jessica, who was the vice chancellor, had about 40 students assembled in a classroom, and I pulled out the talk that I'd given down at Oregon State for their education research council they had a program just a few months before. That talk that had taken 1 hour there, or somewhat less, took 2 hours here because I had so many questions and so much excitement. And there seemed to be so many people who had never heard of phage and thought, these were really cool. These are my kind of people. And I went on then to Kenya and was able to visit a couple of different places. One of them, Joseph, where are you? You here? Anyhow, one of them, there he is. One of them had a student who I'd been in touch with since 2 years earlier, very occasionally. He had tried to come then, and he had isolated staph phage for his master's degree and used them to treat animals in situations where nobody else that he knew was working with phage. And he had convinced his two advisors who were at the Primate Research Center there, who are also part of our gathering here now, to start being interested in phage. So I found people really jumping at it. And then I stopped to visit my former postdoc in Cairo, and he had a crew of there were over 100 people there for the talk I gave, and over half of them were women. And they were excited. And I wound up with such a high that after visiting Martha briefly, I went on up to the Viruses of Microbes, and I said, we need to have people from Africa. Janet here is the only one who was here from Africa 2 years ago. And she's from Nigeria, one of the people here again. So I said that a few years earlier, realizing we had nobody from South America, I had made an offer that from each lab I would give at least one or two free registration fees. And 16 people came from Africa that year, and several of them are from labs that are still represented. And then, and afterwards, the fellow from the Gates Foundation came up and offered to help me pay the flight fees for those people to come. So special thanks to the Gates Foundation, and I would turn this over to Martha.
I have one quick minor announcement to make. So as you may have noticed, the sessions are being videotaped and they are being recorded. And one thing we would like to ask all of the speakers is if you are able to and are willing to sign a release form so that we could release the talk to our YouTube channel after the conference so that we can share this as broadly as possible, because we have many colleagues and friends who for various reasons weren't able to attend, and especially with the types and the quality of the talks at this meeting especially, we would very much like to share those as broadly as possible. So if you are willing and you are on the speaking docket, we have release forms for you to sign for them to be released by our YouTube channel. Thank you. So, could you pull up here? And would you prefer the lectern?
Okay, so thank you very much, Betty, for inviting us all to another beautiful Evergreen conference. Even the weather has turned out significantly nicer than it is in England at the moment. So Betty asked me to say a few words about my recent adventures in Uganda. But I've actually paraphrased my talk with a title, The Spirit of Evergreen. So I'm based at the University of Leicester. I've been coming to these Evergreen phage meetings since 2003. I missed one because I was too pregnant to come, but apart from that, I've been to everyone. And what distinguishes them from most other meetings that I attend regularly is the amount of opportunities we have to speak with each other and to forge collaborations and different scientific partnerships. And this is partly due to the wonderful physical environment that's been alluded to, and partly due to the ethos of the college, and particularly Betty's warmth and generosity in bringing us all together like this. And I wanted to say that my adventures in Uganda, which I'll go on to talk about in a second, are sort of rooted in this sense of sort of collaboration and openness. And many of my, when I first came here as a young postdoc, I remember very clearly in my very first meeting I had a poster explaining a genome that I just sequenced, and I didn't know very much about it. And it was on the cheese and wine night. I put my poster there, and I never made it to the cheese and wine because everyone that walked by gave me a lecture about the gene that they'd worked on for 20 or more years, and I learned a huge amount about my own virus in this. And over the years, I've worked and been lucky enough to work in different people's labs, and then more recently now, I've had my own lab for 10 years now. I've been lucky enough to host many different students. And some of my most exciting and interesting projects have come from collaborations that started here. So I wanted to tell you about a couple of those. And mainly because I looked around when Betty said, who's coming here for the first time? And there's many people, which is great. And I wanted to encourage everybody to not feel shy about speaking to whoever you like, because a lot of my, as I said, a lot of these two examples I'm about to show you started off with conversations over a cup of coffee or beer or peppermint tea. So, Uganda. So first of all, I'm going to, Betty just briefly mentioned Toby, who is here. Oops, she was there, who's here. So she founded a charity called Phages for Global Health. And part of the mission, she'll tell you a lot more about this tomorrow, but part of the mission of this charity is to try to teach people who live in places where phages could be very useful in many purposes, but there isn't the infrastructure or the education to basically work with phages. So part of the mission is to increase the education amongst academics in countries who'd like to use phages. So that's what I've been enjoyed being a part of. So that's Toby and I enjoying, it's because Betty said to say adventures. So we had a nice dancing adventure in Uganda, and also we went to the source of the Nile while we were there. So that's a nice picture of where the Nile starts in Lake Victoria, and that's myself with many of our delegates that we had on this conference. So just to tell you about a couple of the projects that started here in Evergreen. First of all, I want to introduce you to Guillermo Rangal. He is He is a PhD student now in my lab. I met him here 4 years ago. He'd worked in Colombia in Martha Vivas' lab on Pseudomonas phages, and he said, "Oh, I like what you're doing in your lab. I'd like to be interested in doing a PhD." So we managed to find some funding. I think the Colombian government had a relationship with British universities, whereby the universities dropped the fees and the Colombian government paid the stipend. So obviously, not all relationships can just happen overnight. Funding is always an issue. But we got funding, and this is Guillermo here presenting his work on Streptococcus pneumonia phages. This is last year at the Liverpool Viruses of Microbes conference that I helped to host. Now, I would like to introduce you to the second project that started here in Evergreen. So this is Guillermo introducing Nathan, who is clearly really enthralled with the seminars that we have at Leicester. Actually, he's normally a lot more active, but I thought he would probably kill me if I did this. So he was also, he spent his PhD working in Oregon on the impact of viruses on lake communities. And he'd got really interested, partially or mainly through his relationships with Betty, and how could he use these techniques to study phage therapy and look at the impact of phages on the gut microbiota. So he said he'd like to come and do this in Leicester, so we managed to get some funding from the Gates Foundation to do this. So sometimes he's more awake. You can see this is this is him with looking slightly Roman in Leicester. And the other person on the other side was somebody else who I met here 2 years ago. This is Mahmouda Akhtar. Unfortunately, neither of them are here. They're both completely involved in the project that they're working on with Shigella-phages. So we got some funding originally from the Gates Foundation to initiate this project, and then more recently we've just got funding from the Wellcome Trust, to support both Mahmouda in Bangladesh and Nathan in Leicester. And we have a very nice collaboration also with someone from the Gambia. So we've got a large collection of phages and Shigella strains. We have an animal model that we're about to do the first experiments. So the two, and I should show actually, Nathan is not normally looking like that or asleep. He's normally highly active and busy in the lab. And this is when Mahmouda came earlier on this year and spent some weeks in my lab learning how to do some of the techniques on this joint project. So over the years, I've had lots of people who've come through my lab and learned how to work with phages. So when Toby asked me, well, would you be interested in teaching and sort of upscaling this effort and coming to help me with a phage workshop in Uganda, I hesitated briefly and said, more or less said yes straight away, because I'm very fond of East Africa, and I thought it'd be a nice experience, and indeed it was. It was great fun. This is our host, Jessica, who's here. So she and Toby had chatted for a long time, and she set up all the infrastructure that was necessary to teach people about phages. And our delegates, we had 25 participants. Half of them were already academics, vets, doctors, people interested in food security, microbiologists from different disciplines, and the other half were postdocs and students of different levels. So it was a really nice dynamic atmosphere. And we'll hear more about what we did tomorrow. Some of our participants are going to tell you what we taught them. So this is more to introduce you to the context and why and how this came about. And of course to the people, yeah, of course too. So that's so you'll see some. So actually, this is in terms of the people. That's Jessica, as I said. Now this is everybody who was still survived on the last day. Can you see this rather nice phage that Makerere University keeps its water in at the back of the building? So you can see here. So several of our delegates are actually here. So Janet, who is has worked with me in Leicester for about 8 years, so a long time. She's been really. She's going to give a talk later on in this conference about her work on Clostridium difficile. So the two of us, together with Ben, who is, where is he on this picture? There, here. Ben Chun from Yale. We put together a course, which you'll hear about tomorrow in more detail as to what it was. But essentially, it was to teach people how to do all the techniques that's necessary to actually work with phages. But instead of lecturing and telling people you need to do this, this, and this, we did it. We had lectures in the morning, and we had a fully hands-on course on how to work with phages. And the idea really, the ultimate aim was to really hope that we can seed people who have academic positions in different universities. So we had people there from Kenya, Uganda, Tanzania, and the hope was that they will be able to start their own projects on whatever they wish. So this is an example of a project where I helped to train somebody. 2 years ago, I had somebody came to my lab for 1 year called Mahananda Chutia from Assam in India. And the title here, this was the University of Leicester's press office title. So he basically works in the silk industry, and the moths that make the silk are highly susceptible to infection for a large number of different bacteria, mainly Pseudomonas and other gram-negative bacteria. So the poor moths are in Assam they make tea and they make silk, and they put loads of insecticides on the tea plants, and the caterpillars become quite susceptible to infection. So he is really interested in using phages in this context. So he came to the lab and he worked. We taught him how to work with phages. He's now gone back to Assam and he's set up his own research group specifically for the bacteria that are problematic in Assam. And just to show you that it's not wholly altruistic. So we have, because Mahananda needed to work on butterflies, on the moths, we actually then got the Galeria. This is Galeria caterpillar moths. It's an infection model, but we'd never used it in our lab. Because I wanted to show him how to work with phages and insects, we started working with Galeria, and it's been a really useful model. So Mahananda worked with us to get some nice data, which Janet will talk about some of this data in her talk tomorrow. So, because we couldn't work on, these are his moths that he's trying to treat, but they don't even grow inside in Assam. They have very specific conditions. So he came to Leicester, and now he's gone back to do that. So as I said, this is the objective of the course that we ran, was to try to teach people how to do the basic techniques of how to isolate phages, make them clonal, make stocks, titer, and characterize them. So when I showed Anisha, who's also here from my lab, this picture on the plane, she's laughing, because this is how, if you walk into my lab at any random point, you'll see my students. So we spend a lot of our time looking up, is it a plaque? So we spent a lot of time like this on our course. There's a couple of students searching for plaques. So as well as isolating phages from scratch, which we did for both E. coli and Pseudomonas, we took phages with us for a model system, and we went through these other techniques. So we worked on how we looked at the other sort of things, such as probing the interactions between phages, looking at phages, not just looking at phages in vitro, but a little bit more complicated systems such as biofilms. And we also did some molecular biology and started off to do some bioinformatics. And obviously, as half of our delegates are experienced biologists in other contexts, so it was great. We also had a lot of input from them, and an example of that was was Erastus, who has spent a long time looking at different projects in quite sensitive cultural settings. So we learned a lot from him as he talked about the importance of pitching a project correctly, so you don't you bring communities along with you. So that was very enjoyable. So this is you'll meet Atunga tomorrow. He's going to tell you about his experiences from his perspective. And this was and Jelani. Jelani has featured actually on some of my other pictures, but he'll show you in more detail tomorrow. This is hopefully an introduction to who our African contingents are. Yeah, there's, and what we did. So it's very good timing for them. They now have a nice background in pages to learn about everything that everyone's going to present to them. So just want to finish with, obviously we actually did 12-hour days for 10 days, and our delegates never complained about our slightly over-ambitious scheduling. They were with us all the time. The only thing that hurt actually at the end of this course was my laughing muscles for having so much fun. But we had to obviously to sustain ourselves during this period. And being Scottish, I'm very fond of mashed potato, but the Ugandan version is matoki, which you can see here. It's a green banana that's steamed in the leaves. This is how it's sold, and this is how it's prepared. And I became instantly a matoki addict and enjoyed it about twice a day. So as well as all of the sciences we exchanged, we had it was enjoyable and productive time. So anyway, thank you very much for your attention.
Would the African delegation please stand up? And by the way, I should mention that it wound up that the Gates Foundation also helped support that training program, and I think it's really to be hoped that what's happening is that they're getting seriously interested in that whole set of things happening. So welcome, and talk with them and build bridges. So we're very lucky to have our next speaker. That's Jean-Paul Pernet. And I learned just about a month and a half ago that actually one of my biggest dreams in terms of phage work is in the process of coming true. In general, at most of these meetings, there have been very, very few talks about actual phage therapy. And that's partly because the few things that were happening were very complex, company trying to do work that was leading towards eventually patenting and all kinds of things, double-blind experiments. Yet it's been used so much in many parts of the world. And Jean-Paul has led the group in Belgium that for a number of years has been trying to work out the same sort of thing we're interested in, and that's developing ways of giving people ready access to phage therapy as it's practiced and has been practiced elsewhere, and working through the governmental obstacles. And there's a group also called Phage Biotics that has grown up, that's a major part of it in the Belgium area. And I'd like to turn this over right away to Jean-Paul. And he at first thought there was no way he could come, and finally managed to get permission just a few days ago from the military command there to be here today, and I'd like you to welcome Jean-Paul.
Thank you, Betty, for the invitation. Thank you for having me. This will be my first post-barbecue plus beer plus jet lag presentation, so bear with me. So I'm going to talk about phage therapy research in the Queen Esther Military Hospital. Now in Belgium, the military hospital is known for its burn wound center. It's really dedicated to burn wound patients. We have 8 ICU rooms, 18 medium care rooms, and over 10,000 consultations per year. Okay. But it's also known for its medical response to mass casualties. And we were able to put out 80 beds in 20 minutes with equipment. And unfortunately, the last time we had to do this was last year after the terrorist attacks in Brussels Airport and in Brussels Metro Station. And this is the kind of wounds we used to see then. They are similar to battlefield casualties. They are burns, open fractures, penetrating injuries, and you can easily understand that they get infected often. Luckily, thanks to medical advances, we are able to save even very desperate patients. This is an example, a 9-year-old boy burned for 95% of his body surface. Okay, we use classical techniques like excision of burn tissue as soon as possible. And then ideally we would cover with autograft, so with the skin of the patient, which we would enlarge using different techniques. But unfortunately sometimes there are insufficient donor sites. You only have like a few % to work with. And often also there's infection, so you have to wait. And the wound bed often is also not ready to accept the autograft. And then we use donor skin, a skin from a deceased person. And then we also use some what they call advanced therapies. For instance, we will use neonatal foreskin keratinocytes, or we will culture the keratinocytes or other skin cells of the patient himself or herself. And so these medical advances have led to improved outcomes for those severely injured patients. But today, most die of bacterial sepsis. And so the antimicrobial resistance crisis aggravates this situation. And you also must know that military medicine historically involved the prevention and treatment of infectious diseases. And also phages were used in military medicine. For instance, by the Red Army in the Finnish campaign, or the Second World War, by the German Army, by the British Army, Japanese Army. So in 2003, we also wanted to research phage therapy. But as always, beginnings are difficult. So what you see here is like a scoring of the proposals in 2003. And you can see that our proposal did very bad. So we were not the last ones. But it was dismissed as being like science fiction in 2003. And we were lucky to end just before collecting postcards featuring military uniforms pre-Waterloo 1815. So that's why we decided to find some strong partners, to find some strong collaborations, national with the University of Ghent, Professor Mario van Eekhouten, with the University of Leuven, Rob Lavigne, and also with their networks, for instance, with the University of Guelph, with Andrew Kropinski, but also international collaborations with the Eliav Institute. And I'm quite sure that Queen Estrid and George Eliava would have approved this collaboration. They both lived in the first part of the 20th century. And I would also like to promote a poster of two researchers from Eliav Institute, Elena and Katuna, who unfortunately could not make it here due to visa problems. Other collaborations with University of Exeter, with Professor Buckling, with DSMZ in Germany, Christine Rode, and with Institut Pasteur, Laurent Debarbieu. Also interactions with Phages for Global Health, with Toby Nagel, and also want to promote a poster of Alice Maestri on Vibrio cholera phages. And also nice interactions with Betty, who also visited the military hospital. So today I'm happy to say that we have strong phage therapy research in the military hospital, and we have 6 branches. So first, but very important, is that you have to know your enemy. So you have to study your target bacteria. You have to determine the bacterial populations. And the target bacteria we are interested in are the usual culprits, Pseudomonas, Staphylococci, Acinetobacter baumannii, and recently also Klebsiella pneumoniae. And we have a collection of more than 8,000 strains. And this is just an example of what population structure can look like. And this is the one of Pseudomonas aeruginosa. And there we discovered that most problems we had were with the MDR012 clone. So we for sure had to target that clone. Next step is, of course, you need to have good phages, adequate therapeutic phages. So we have to isolate and select phages. And you might know that in Belgium we know something about developing beers, but phage cocktails is not our specialty. So we went to Georgia, and yeah, this is not working. I don't know why. Yeah, but then, first part it worked, but then Dr. Maya Meabishvili agreed to come to Belgium, and for now 6 or 7 years she is working with us to develop phage cocktails against those target bacteria, of course. But next to that, she also developed phages against E. coli strain 0104H4 that caused the outbreak in Germany, and she's now working on mycobacteria. Then, of course, we also wanted to study the bacterium-phage interactions, bacterium-phage coevolution, and with an emphasis on resistance evolution. What do we know about the host-parasite relationship? Well, we know it's complicated. So phages will not eradicate their hosts. This is logic. So the patient's immune system or other antimicrobials need to eradicate the bacteria. And so to evade the phages, bacteria quickly develop resistance to phages. But new phages can emerge to infect these resistant bacteria. So it's an arms race. What do we know about phage resistance evolution? Well, we know that it will eventually always emerge, in vitro at least, even to cocktails. And then it might also be better to apply phages sequentially instead of all together in one cocktail. And using that strategy in vitro, we found that we could we could prevent bacterial resistance from emerging. So when you use them one by one, bacteria don't have the time to develop resistance. Once they start, they are hit by another phage. So that's the rationale. We also know that you would like to pre-adapt those phages, because we know that pre-adapting phages, or training them, like they say in the Java Institute, will lead to an increased pathogen clearance and to lower resistance evolution. We also know that mutations that confer resistance to phages in vitro come at a certain physiological cost. But little is known about phage resistance evolution in vivo. Yet, phage products are on the market for decontamination of food. Phage probiotics and products for farms are in development. But what do we know about the impact on bacterial resistance to phages? What about the impact on bacterial populations in the environment? Shouldn't we study this first? I don't say I'm not against the use of phages in agriculture and fisheries, but I'm just saying that now that we are starting to do that, why not look at resistance evolution in the beginning? So this leads to the burning question: should phages be produced, marketed, and used as if they were antibiotics? This means broad-spectrum phage cocktails used empirically without limitations in agriculture, fisheries, and food. So this brings us to the regulatory branch. In the EU, phages are medicinal products. That's a fact. And this means that we need to follow the conventional medicinal product licensing pathways. They should be manufactured according to GMP. We have to have preclinical studies, to have phase 1, 2, 3 clinical trials, and marketing. But these pathways have been developed for fixed drugs, such as antibiotics or aspirins. And they are less suitable for flexible phage therapy approaches. They also require a lot of money and a lot of time. And do we have that time and that money? So it was estimated that in 2050, 10 million patients will die annually due to multi-resistant infections, and most of them in low or middle income countries. We also know that large investments require strong IP protection. But phage therapy is in the public domain since the 1920s. And we also know from the Myriad Genetics case that it's unlikely to patent products of nature. But we think that pragmatic solutions are possible. And there's an example, the fecal microbiota transplantation. So how does it work? You have a patient, typically with Clostridium difficile diarrhea. You would find a donor. You would harvest the feces, dissolve them in saline, filter or mix them, and then give them to the patient. And according to some studies, close to 100% of success. So fecal transplantation and phage therapy are both facing same problems of public perception issues. Fecal slurry contains billions of bacteria, but also billions of phages. Yet, they have not been classified as medicinal products in the EU, and this means that they do not need to be produced according to GMP, and no marketing authorization is required. So the MDs just went along doing their trials, and they did some good trials, and they were able to show efficacy. And it has become an established practice in the EU. For instance, in Belgium, we have recommendations regarding the therapeutic indications, the procedures, the safety, and the quality of the transplantation of fecal material. This is an official document of the Superior Health Council. And I'm happy to tell that also for phages we have now a solution, a pragmatic solution, because on July 5, 2016, the Minister of Public Health acknowledged that it is difficult to determine whether phage preparations should be considered as industrially prepared medicinal products or as magistral preparations. And in the US, that would be the equivalent of compounding pharmaceuticals. And these are prepared in pharmacies officina, can be the pharmacy in the street, and they are not subjected to constraints related to marketing authorization. And in October of that same year, it was formally agreed that natural phages whose derivative finished products are not fully compliant with the requirements of Directive 2001/83 relating to medicinal products for human use and for which there is no monograph in official pharmacopoeia can be processed by pharmacists as active ingredients in magistral preparations. Now, this brings us to the fifth branch, the production of those phage preparations. Because MDs want to use phages. The problem is that they are not able to find approved phages. So in 2007 we started producing our own phage preparations, and we used our experience in cell therapies and production of cell cultures to produce those phage preparations. And now we are moving towards the production of magistral phage preparations. How does it work? First part in the laboratory, you would select characterized phages from a phage bank. These would be selected against a pathogen. Then you would go to a clean room, or better, a clean room environment, because it does not have to be necessarily a clean room. And then there you would produce your active pharmaceutical ingredients, API, and that would be a single phage produced in a suitable host according to a monograph, which includes QC tests, so quality control testing. And then these APIs would go to the pharmacy, where the pharmacist would produce a magistral preparation upon prescription by an MD. And this preparation could contain one or more phage APIs in a carrier, which could be hydrogel, for instance. And this is the monograph. I will not go into detail, but it describes production, also the equipment you can use, the production environment, the tests, shelf life, labeling. We'll quickly focus on the QC tests. Of course, you would have to test your host bacterium. You would have to identify it, but just using classical clinical microbiology techniques. You have to check for lysogeny. Although you would not want to have a lysogenic phage, if you cannot find another one, you would be allowed to use it. Lysogenic bacterium, excuse me. Then also you have to test your phage seed lots. You would have to identify your phage using DNA or RNA sequencing and genome analysis. You would also have to screen for genetic determinants conferring toxicity, virulence, lysogeny, and antibiotic resistance. You have to test your phage APIs, phage identification and quantification, but here at this level you would not have to do sequencing. You would be allowed to do a simple specific qPCR. And then you have to determine your bioburden, and it should be less than 10 colony forming units per 100 milliliter or per gram. And of course, for IV use, it should be sterile. You have to quantify your bacterial endotoxins using a LAL test, determine the pH, and if the phages have been freeze-dried, you have to determine the water content, and if you used chloroform in the production, you should test for residual chloroform. We also looked for the stability in wound care products. This paper came out recently. I will not go into details, but some wound care products mix greatly with phages, others don't. So you have to check for this. The production concept, it's a closed system, small scale, ad hoc. Only host bacterium and phage seeds are inserted. Purified phage comes out 24 hours later. Air inlet and outlet over filters. One use, with the exception of the pump and the incubator. No need for clean room environment. This is how it looks like. I will not go into detail. Did some testing in collaboration with PAL Life Sciences. We took PNM phage from Ilyava Institute. We tested 24 combinations of filters, bioburden filters, chromatography capsules, and buffers. We tested pressures in the system, phage yield and titer, and the toxin level, pH, and the winner produced 1 to 2 liter of log9 PFU in PBS, which would give 1 to 200 liters of log7 per milliliter, which is the concentration you would use normally in patients. And endotoxin levels are compatible with IV application, pH 7. This is just some pictures illustrating optimization. And this is the picture of the clean rooms. One of the 4 clean room suites has been accommodated for phage production. So finally, last branch, clinical applications. A disclaimer, I'm not a medical doctor. I'm basic education biotechnology engineer, but sometimes medical doctors need engineers. You all know about that the European Medicine Agency is at a workshop dedicated on phage therapy in London. And the conclusion was that we need randomized clinical trials, which is, of course, the holy grail of medical research. And the European Commission funded a study, a phage therapy study, Phagoburn, which is phages against Pseudomonas and E. coli infections in burn patients. 2 companies produced the phages, and then there were 3 centers in France, Switzerland, and Belgium that coordinated the clinical trials. The trial was completed and blinded in June 2017. The analysis is still ongoing. I'm not allowed to tell you anything about the results. But some issues were already highlighted in science report. So the manufacturing of one batch of the investigational product took 20 months and the largest part of the study budget. And then physicians were reluctant to include patients because the study product targeted only 1 bacterial species, and burn wounds are often colonized by multiple bacterial species. We did our own pilot clinical trial in 2008 because then phages were not yet classified as medicinal products. We were able to do this under the umbrella of the ethical committee, but we only allowed 10 applications and only 5 milliliters of phage cocktail, so we were not able to show efficacy, only that we had no adverse events. And from then on, we treated patients under the umbrella of Article 37 of the Declaration of Helsinki. And this is where proven therapeutic methods do not exist or have been ineffective. A physician must be free to use unproven or new therapeutic measures if, in the physician's judgment, it offers hope of saving life, reestablishing health, or alleviating suffering. So under that umbrella, 12 patients were treated. I will just show the most spectacular one. There was a 62-year-old man with diaphragmatic hernia with bowel strangulation. He developed gangrene, leading to amputation of the lower limbs and fingers. He had large infected pressure sores, and he did chronic septicemia with extensively drug-resistant Pseudomonas aeruginosa. He was transferred to the military hospital in October 2016 for surgery, and he was put on colistin, which was the last resort antibiotic at that time. So what happened is, after 14 days of colistin, you can see that his kidneys started to fail, which is not abnormal with colistin, which is very toxic. You can see the creatinine and the ureum levels rising in his blood. So at that point, the clinicians decided to stop colistin administration. And then what happened, what could be expected, was that the patient did bacteremia with Pseudomonas aeruginosa. So you can see temperature rising, CRP levels, CRP is C-reactive protein levels rising, and 7 consecutive positive hemocultures in 3 days. So at that point also the patient was in a coma, and in consultation with the family, the physicians decided to start phage therapy with Pseudomonas aeruginosa phages. In fact, it was a cocktail that also contained the Staph aureus phage. And this is a result of 10 days of phage therapy. So all parameters returned to normal, and his kidneys had the chance to recover. You can find more details in the case report. And this is like very new results I got a few days ago, so it's very preliminary. We checked for the immune reaction to those phages, and so there was an application on the 13th of November, and it's like, well, first I've explained what we are seeing. We see the neutralization of the phage by antibodies. The red one is phage 14-1 from Moscow. The green one is phage PNM from Ilyava Institute. The blue one is phage ISP, also from Ilyava Institute. And what we see is that after 8 days, we get neutralization, quick neutralization of phage 14-1. And it's like 4 or 5 days later that neutralization of phage PNM starts. But for any neutralization of phage ISP, we have to wait for a second application of phages, which happened several months later. And and maybe it's not a coincidence, because phage ISP, it stands for intravenous Staphylococcus phage. And so maybe this is why it's such a good intravenous phage, because it takes even a second application to have neutralization of this phage. So I want to thank my phage colleagues at the military hospital, and I also want to thank you for the attention.
So you can.
I'm going to bring up, okay, great.
Good. All right, so we have some time for some questions now. If you would like to ask a question, please raise your hand and I will bring the microphone to you. If we have a question that doesn't get onto the microphone, I would ask Jean-Paul to please repeat the question so that the cameras can hear it as well. Thank you. Questions?
Hi. I just had a question about your the phage neutralization, one of the last slides you showed. Is that the appearance of neutralizing antibodies in vitro? So this is, what you're seeing is the appearance of basically ability to block plaquing, is that correct?
Yeah, this is the results of mixing dilutions of serum samples with phage.
Okay.
And so...
So we don't know necessarily that the clinical benefit has been abrogated by these antibodies, is that correct?
Yeah, it's in vitro testing, yeah.
Okay, thank you.
Hi, I was just wondering how different is the ISP phage from, like sequence-wise, from the other two phage?
I think they are quite different because ISP is a Staphylococcus aureus phage, and the other two are Pseudomonas aeruginosa phages. So I think. Well, PNM is a podovirus, ISP and 14-1 are both myoviruses. So, but I.
They have no DNA homology whatsoever.
No. So this shows that, at least in vitro, that reaction towards well, in vitro, like the sera came from the patient, so it's not like we like the antibodies were made in the patient, but that the immune reaction seems to be quite different according to different phages. So this is important when we would like to use phages intravenously, to use phages that do not elicit immune reaction after a few days.
Jean-Paul, I have a question about the burn patients that you worked on. Were you using the phage both intravenously and topically? And if so, what frequency were you doing? Like how often in a day were they being applied?
Yeah. Well, for application of phages, the physicians mostly use the protocols that they use also in Eliave Institute, that you also can find in some literature. And it would be, ideally it would be like a few times per day even, but like in burn wounds it would be difficult, so it would be one time per day. But it's important that you first neutralize the wound, that you first rinse with buffer, for instance, because the phages don't like an acid environment or basic environment created by the bacteria.
But did you also use intravenous with them? Besides topical, did you also do intravenous?
Well, we did one patient intravenously, and that's the one case I showed. Yeah.
So then, Jean-Paul, I wanted to get a better understanding about this. Does this correlate with the clinical experience, this neutralization in vitro data? How does that correlate with your clinical experience?
Well, yeah, it's a good question, but I think the clinical part, as you can see here, is that the phages worked, and so they were used for 10 days. And when you see at the graph, like the one phage, you would have like an immune reaction after 8 days, and the other one after 12 days. So And also, must not forget, I didn't tell this, but those phages were not given as a bolus, not once in one time. They were given like in 8 hours. It was 50 milliliters during 8 hours every day. So I think that during 10 days, this cocktail should have worked. And when you look at results, it at least protected the patient from further bacteremia during those 10 days. And it was important because his kidneys could recover during those 10 days without antibiotics.
I have a question. Hi. Did you happen to culture the Pseudomonas from that patient?
Yes.
And did you do any resistance development studies in vitro?
No. So of course, we tested the activity of the phages, and both phages were active against that strain. But of course, it's difficult because, as you can see, immediately upon application of the phages, we had no Pseudomonas aeruginosa anymore. So we would have loved to look at phage resistance, but we don't have any bacteria in the blood after the administration of the phages.
Yeah, as a follow-on to that question, it'd be interesting to know whether you could actually culture out the bacteria from the wound bed, from the burns, and see if those bacteria are in fact resistant to the phage.
Yeah, that we can do. Yeah, we have kept the bacteria from the wound bed, so we can have a look at this. We're also planning on doing qPCR on the serum samples to look what the phage does, but this is very preliminary. So.
All right, thank you very much.
Thank you.
Our next presenter is also talking about clinical experience in quite a different way. Naomi Hoyle is the person who works quite a lot with patients from outside the country at the Eliava Institute. She's a junior physician there. She's also an Evergreen grad. Did her medical studies in Georgia.
Thanks. It's great to be here and to be actually on the lecturer stage at Evergreen. So as a student, a former student of Evergreen, it's great to be back. Nice to meet the president, and great to see all of you. And this is an amazing turnout for an Evergreen meeting. So I am Naomi Hoyle. I'm from the Eliava Phage Therapy Center, and this is kind of a summary of some of the clinical work that's been going on at our clinic. I am actually the patient coordinator for the clinic. The clinical cases that I'm going to present are actually led by physicians at our clinic, so they're not my personal cases. Our clinic actually is unique in that it has specialists on site and is really a team approach to each patient. So we have a urology department, pediatrics, internal medicine, ENT, gynecology, surgery, and then we have specialists in pulmonology, in dermatology, endocrinology, and all of those people are really important for us, and I'll tell you about a little bit more about why. We have a variety of services also that we can provide to our patients. We have the Eliava lab, which is Diagnosis 90, and there we're able to look at phage sensitivity testing to the standard phage preparations that are produced by the Eliava Institute, and also look at what kind of custom phages we're able to do in case of phage resistance. And we also are able to do all of our biochemical testing there and clinical testing. We have CT, MRI, and ultrasound on site, and also physiotherapy and massage therapy, which you might think, what does that have to do with phage therapy? But it does. It's connected to phage therapy. It helps our patients feel good, and that is important to us. So, and then, as I mentioned before, we have a huge library of phage, which is part of the Eliava Institute and gives us the ability to make custom phages for bacteria that are not part of the phage, standard phage cocktails, or for phage-resistant bacteria. Some of the frequently occurring diseases at our clinic include prostatitis, urethritis, chronic wounds, diabetic foot ulcers. We have a lot of gastrointestinal patients. Irritable bowel syndrome is a very interesting topic that I'll talk a little bit about later. We have respiratory system diseases, patients with cystic fibrosis, and we have our superbug infections, which is actually not a disease on its own, but it's kind of a key word for patients that we're able to treat nosocomial infections. As Jean-Paul mentioned, the antibiotic crisis is actually really a big deal and will become more and more of a problem as we go along. It was very encouraging to see that Bacteriophage is part of the White House's initiative on a plan against antibiotic resistance, and the EU also has phage therapy as part of the plan, which is in their action plan against rising threats from antimicrobial resistance. So there is a global stance on bacteriophage being a viable alternative. There's no question about that. The question really is going to be, how are we going to get that available to people? So we've had more and more people coming to our clinic. In the last year, from 2015 to 2016, we had doubled the patient that we were seeing there, and then in this year, 2017, we've already doubled that. So we're really seeing a lot more patients, and that gives us a big opportunity to look at different conditions. We've had a lot of media exposure, and that's been really important in terms of popularizing phage therapy and the idea of being able to treat antibiotic-resistant infections with phage. So we had a piece on Reuters, on Beckman German television, the Dutch TV Zorg, BBC, Norwegian television, and a few other documentaries that have been filmed that will be coming out in Holland and in Germany. Through this popularity, we've also gained a lot of partners who are facilitators of phage therapy, so basically medical tourism companies who are helping to coordinate the travel to Georgia, which just coming over myself with my family, I realized how hard it is for patients being sick to travel. So this is a big help for them. This is C'est Soigner Georgie. There's a new company that is US-based called PhageRx. And we have a new Spanish partner, Indian partner, and partner from the Netherlands that are also going to be coming out soon. Oops, what did I do? Okay. So when we have a patient contacting us, the first thing that we want to identify is what is the disease that they have, and is this something that we can treat with phage therapy? So we want to review as many records as possible, and that's an issue depending on where the patient is from. American patients don't generally have that kind of accessibility or the ability to share, or so there are some challenges there, but our goal is to make sure that we're screening for patients who are eligible for phage therapy, and then we'll do initial testing of samples to see sensitivity to the standard phages, or if they need to have a custom phage made, which takes an additional amount of time. So once the patient has arrived at our clinic, we do another detailed history, which can often be and uncover a lot of things that we didn't know previously about the patient. We'll repeat our sensitivity testing, or we might be doing it for the first time if they weren't able to send a sample in advance. We'll do other diagnostics because our main goal here is to identify and diagnose the patient, identify what is the main problem, what is the main disease. And a lot of patients really haven't had clear communication with their physician, so they might even have an infection that's antibiotic sensitive, but they haven't been able to get the care that they needed at their home clinics. So we're doing a lot of diagnostics. And our team meets collaboratively, so each specialist will see the patient and then have some collaboration to determine what is the treatment plan and what is the strategy for each patient. If a patient needs to have a custom phage made, that's something that we want to know in advance because there's a 6 to 8 week lead time because the phage needs to be developed first, and then, of course, quality control and sterility needs to be done. So this is a list of the phage preparations that are produced by the Eliava. These are the standard phage preparations. I know some people were interested in knowing what phage cocktail contains what phage against what bacteria. So we have Piophage, Cessphage, Pharisees, Enco, and Intesti, and the Staphylococcus phage. And then we also have the ability to do those custom phages. So a really important question that we've been asking as physicians is how can we assess improvement? We have a great institute, the Eliava Institute, which is doing a lot of basic research and also doing clinical research, but when we're starting to use phage in the clinic, we need to assess it in a different way. So how do we assess what are the main endpoints that we're hoping to find that we can assess improvement with? The first thing that we've come up with is really just the patient's subjective criteria. And this is something that probably scientists are probably cringing about because we're not able to really quantify that in a very scientific way. But that's important for us because our first goal is that the patient has a curative effect, that they are able to resolve their infection and to get better. Apart from that, we look at as much objective criteria as we can get, and I'll talk a little bit about that more specifically in some of the case studies that I'm going to present. Bacterial titers are really important, looking at before and after, and even in between, and looking at the progress. As well as whatever laboratory clinical testing we can do, looking at CRP, although in some chronic infections we don't have a big increase in CRP. We might look at comparative ultrasound or comparative MRI, trying to get proof, basically, of this efficacy, and that supports our patients. What they're telling us, which is that they're feeling better. So now I'm going to talk about a few case reports, looking at cystic fibrosis, phage in non-healing wounds, phage as preventative medicine, and just one brief case on urology. So cystic fibrosis is something that's been studied at the Eliava for many years, and this actually started through a collaboration with Evergreen, and was one of the things that I was working on as an undergraduate student, was looking at phage sensitivity for cystic fibrosis strains that, or strains isolated from cystic fibrosis patients. So we're looking at, there's a huge problem with the emergence of antibiotic resistance in cystic fibrosis patients because they have a genetic disease. They need to take antibiotics basically all the time, and they need an alternative for the infectious control. So our approach is to continue the standard treatment regimen for CF, which is based on national guidelines. That is to have them on antibiotics, on enzymes, pancreatic enzymes, bronchodilators, mucolytics, whatever it is that they were coming to us with already prescribed. Here I'm going to show you about 3 different cases. So this is kind of an overview of the last 2 years of CF patients who has been treated at our clinic. So for the most part, we have pulmonary cases of CF. It's possible to have also mixed form, and most of the patients have had CF since, or had a manifestation of CF since childhood. The average age of our patients is 24 years. So the general trend and one of the main criteria that we're finding to be as an endpoint or as evidence of improvement is the number of hospitalizations for IV antibiotics. And the general trend is that all of our patients who were going into the hospital for at least two to 4 times a year for IV antibiotics was greatly reduced. And I'll tell you about 3 different cases of specific patients. One is of a 44-year-old female. She had a late onset of symptoms of CF, and her main symptoms were cough, generalized weakness, and she had MDR Staph aureus, as well as Pseudomonas infections in the past. At that time, she didn't have Pseudomonas infection. She was given oral treatment via nebulizer, and we used 3 different phage preparations with her. And the results of the microbiology was quite impressive. We had a decrease in the bacterial load from 10 to the 6th to 10 to the 4th after 1 week, and then less than 10 to the 3rd after 2 weeks. Her general condition greatly improved. She had the oxygen saturation also improved from 92 to 96%. She had improvement by auscultation, so just listening to her lungs sounded a lot better, and less dyspnea, so she was breathing easily. She gained weight, and then decided with her doctor to discontinue the antibiotic that she was on. And this is kind of an issue with CF patients because national centers want them to be on the specific guidelines, and so we also want to make sure that any decision that's made about taking away antibiotics is done together with their doctors, who are their primary care physicians. At a 6-month follow-up, she continued to be doing really well. She'd done 2 treatment courses in 6 months, so she does take phage periodically via nebulizer, and she had gained 5 kilos by then, so that's another endpoint. She hadn't been hospitalized at all, whereas in the past year she'd been hospitalized 3 times for IV antibiotics, so it's a significant improvement in her overall condition. A similar case in a 10-year-old girl from France who came to us actually with acute pneumonia. We had to hospitalize her in the ICU of the Children's Hospital in Georgia, and she was started on intravenous antibiotics. Her condition was quite severe at that time, but as we were working with the ICU doctors, we also added the phage preparation to her treatment plan, which was vancomycin meropenem, and she had been taking that for 4 days, and after the addition of the phage, she started to get better. So it's questionable if this was just a coincidence that it took this long for the antibiotics to start working, but we feel that it was very helpful to have the phage added to that, and this was a phage that we knew to be sensitive to her strain. So her oxygen saturation went from 87% to 96% after this, and she continued to take the phage periodically. She developed resistance to the phage that she was taking, and we had a custom phage made for her. We just saw her a week ago, and this was a 12-month follow-up. So after 1 year, she hadn't been hospitalized a single time during that year, whereas she'd been hospitalized more than 5 times in the previous year. She was doing great, and what was most exciting was that her lung function had improved significantly. So it was 48% the previous year and is now 67%. So it's another marker that we're hoping to be able to get more consistently to see lung function before and after using spirometry. And the third CF case that I want to tell you about is an interesting case of a patient who started having lung history from 11. She came to us when she was 17 with MDR Achromobacter, and we didn't have phages for that, so the DSMZ kindly donated phages that were made into a phage cocktail for this patient. When we gave the phage to her, she also dramatically improved, and her lung function also dramatically improved. It was 58% when she started, and it improved to 69%. The 6-month follow-up also, she didn't have IV antibiotics. In this case, she did have her normal scheduled IV antibiotics, but when she followed it up with phage, then her lung function further increased to 84%. So this young woman was a dancer, and before treatment, she was not able to dance, and now she's out dancing again, and that was her main goal. So that's really exciting to have such a restoration of lung function. I want to just now skip over to some of our surgical patients and talking about non-healing wounds. This is a patient with chronic osteomyelitis, and chronic osteomyelitis is a really challenging case to take on because of the chance of recurrence. We originally wanted to have this patient do operative intervention, and she refused. So we decided to try at least phage therapy before amputation as a last result for her. And over a 2-month period, you can see in the photos the dramatic improvement of her case. This is a new case of a diabetic foot ulcer. We're really interested in doing more of the diabetic feet. We've had several cases which have been very positive in terms of results. And this is just looking at a 1-week progress of treatment. So this is before treatment, and you can kind of see, I don't know how well it shows up on the pictures, but it's quite deep in some places. And by the 4th day of treatment, you're already starting to see granulation tissue there. So, and after 1 week of treatment, I'm just counting the days because this is only the 3rd of August. Yeah, this is very new. Yeah, you're seeing the size also decrease. So this is really working for these kinds of cases, and I know we're going to have a whole talk by Randy Fish about diabetic foot ulcers. So I think it's really very interesting and very exciting work. We were approached by the child of a patient, actually grown man, but who wanted to try phage therapy for his father, who had something called a left ventricular assist device. And this is a really unusual kind of device that treats heart failure. It's got an external pump, and this is, well, external meaning outside of the heart, but actually inside of the chest wall. So this pump is put in and actually does the work for the heart. This patient got his pump infected in 2013 with MRSA and has been having problems ever since. So when he contacted us, he was in the ICU, hospitalized for sepsis. This is a picture of the wound of the chest wall. It's a non-healing wound. And his doctors. So he was hospitalized in Germany, and he was flown then to Spain, where his doctors agreed to try phage therapy with him. We tested swabs, but weren't able to identify the pathogen, so we were doing this treatment empirically. But they were using jet lavage to clean out the wound, and then putting a phage-soaked gauze into the wound, leaving it for 4 hours, and then after that doing vac therapy on it. So the results of this are actually difficult to say, because we didn't have the initial bacteria, but this patient has not had a recurrent infection, and the wound was able to be closed and is healed. So that for us is total success. The patient is monitoring his CRP, and anytime that starts to go up, he takes some staph phage, and he'll do a treatment course. So he's done 3 treatment courses now, and he'll take a short course, maybe 10-day course, and the CRP goes down again, and he's feeling great, and they're really very excited about that. So we've had lots of other patients come into the clinic who have some kind of antibiotic-resistant bacteria and are wanting to do a scheduled operation. So they're not allowed into a hospital because of their resistant bacteria, and so they're not able to get their operation done. So we've been able to eradicate ESBL, 3-MRGN, MRSA, MDR, XDR, all of those kinds of bacteria, which are basically the root of nosocomial infections. This is an interesting case also, very dramatic result. The patient was very ill with a recurrent UTI. He had an indwelling catheter, and there was biofilm and catheter-associated infection that was going on. He had Klebsiella, and the problem was that he was having kidney failure, so he was no longer able to take antibiotics, and especially since they didn't work, it didn't make sense to take them. And a custom phage was made for him kind of on an emergency basis, and came out within 4 weeks, which is quite quick for custom phages to be made. And he was treated at home in the Netherlands, and complete eradication. His family asked for his picture to be shown in this talk. They're really excited about the results, because basically he was extremely ill. He's also taking phage as a prophylactic now periodically, and kind of gauges when the urine starts to get cloudy. He'll use one of the broad spectrum phage cocktails through his catheter, and finds that that's keeping the infection at bay. So interesting to think about some of the patients that are really using the phage long term, and I'll talk a little bit about that more. How am I doing on time? Okay. So I can't forget our Georgian patients. Bacteriophage in Georgia is famous for intestinal problems, intestinal disturbances. So we have a lot of patients, a lot of Georgian patients with irritable bowel syndrome. We haven't been able to analyze much of the data, but we've analyzed a little bit. Irritable bowel syndrome doesn't have every aspect of the pathophysiology worked out yet, but we have the idea that there's altered GI motility, there's hypersensitivity maybe to bacterial byproducts that's causing the main problems, which is abdominal bloating, pain, and altered bowel movements. So really interesting information is coming out through all of the microbiome work and the metagenomics that we're seeing now emerging. Just to look retrospectively at 52 patients, they had various forms of IBS, diarrhea, constipation, and mixed form. 39 of those patients had E. coli, 3 Klebsiella, 3 Staph, 5 Enterococcus, and 2 Pseudomonas. So these are pathobionts. We are not sure if they're pathogens, but we're finding them in excessive amounts in stool samples. So we're looking at those patients and using different phage preparations to treat them. So we're not using a single phage preparation in this case. We're using it based on the sensitivity testing. We also have a group of patients, which we haven't analyzed yet, which are based on empirical treatment using a single phage preparation. So the results showed that 85% of cases eradicated the pathobionts, but only 75% of them had clinical improvement. So our observations were that the phage was effective in treating those specific bacterial overgrowths. All of the patients had already gone through traditional treatment for IBS without result. So the addition of phage to those patients and treatment regimen is actually what was making them better. So it would be really interesting to look at this more in depth and look at it in another way instead of just classical cultural methods, looking at also metagenomics and looking at the microbiome on a different level and what's going on there. This is a really interesting and very unique case that we had of a patient with Netherton syndrome. Netherton syndrome is a rare genetic disorder, which is a combination of ichthyosis, allergy, and bacterial infections. This patient was 16 when he first came to us, and he had chronic Staphylococcus skin infection. It was a complication of the Netherton syndrome. He has severe allergy to most antibiotics and resistance to the ones that were tolerable, and I have a list of all of the drug allergies to show you next. But basically, he's continually hospitalized for sepsis, and his doctors in France were the ones who encouraged them to come to Georgia because they were afraid that this would be his last chance, that he would not survive these infections. So the list of allergies are huge, unbelievable, basically almost to all antibiotics. Also allergic to corticosteroids, to most topical agents. So we're looking at a skin disorder. We can't use any creams or ointments. Very, very limited there. And so what we did was we used the liquid phage with him and also had a special phage cream made for him using a base that he was not allergic to. And he did not only topical but also oral use of phage. So this was at 90 days of treatment, 180 days of treatment. Basically, within 1 week, though, we were seeing that he was starting to get better. So that was what kind of encouraged us to continue on. But the results were great. The first application of phage was quite suspenseful and scary because we didn't know if he would be allergic to the phage. So we did apply the phage in a very small area first, and he was tolerating that. So then we went on to use the all over the place. So this is a 6-month result. When he first came in, his skin was kind of, well, I don't know how well you could see the first pictures, but there was no normal patches of skin. And here we have almost all the skin is normal. After 1 year, he was just in our clinic last month. He has hair. He has normal skin, and he's so excited. We were talking about basically the last chance before, he would die from sepsis, to talking about what kind of hat he needed to buy to protect his face from the sun. So it's really, really exciting improvement there. This case was published in Frontiers just recently, and that's something that we're also really excited about because one of our goals is to get more cases published and to analyze more of the data that we have, which is a lot of data, because we're doing a lot more treating than research. We would like to participate in well-structured, controlled clinical trials, and that's something that we feel is really important as Western physicians who are practicing evidence-based medicine for the most part. We'd like to improve some of the collaborations and promote phage therapy internationally. So here's our team, which is quite big. The Eliava Institute, the Eliava Foundation, the Eliava Pharmacy, we're the Eliava Phage Therapy Center, and Diagnosis 90. So thank you for your attention.
All right, so to try and keep things a little closer to the schedule, only a couple of questions, but we will be having a poster session immediately after these talks where I encourage you to find Naomi and ask her any more detailed questions you might have. Thank you.
Okay, quite spectacular result. I have a question regarding this last case of 16-year-old boy from France. Did you have only phage therapy performed on him, or you also treated him with Borjomi or something, you know, balneological? Because Georgia is known for being climatic spa space.
Yeah, so we use Borjomi not as an external application, but just because it's a good alkaline solution. So if we don't have Borjomi, we use baking soda and water to neutralize the stomach acid, and otherwise we use Borjomi, which is a Georgian mineral water. One thing I'd like to add also about that patient is that we also are following the neutralizing antibodies for patients. And in his case, he's basically using phage constantly. He does have increased antibodies to the phage that he's using, but we can't say that it is affecting him clinically. The opposite. It seems to have absolutely no effect on his clinical improvement.
Hi. Very nice talk. I wondered if you could just give us your thoughts briefly about endotoxin in your phage preps. I mean, I think we all agree topically it's not a big deal. We have limits for parenteral use, but I don't know at least how to think about endotoxin in the lung with nebulizers. Can you just comment on how you guys see that issue?
So I can just address that from a clinical standpoint because I'm not privy to the laboratory side of that, and we have not seen adverse reactions neither to inhalation or to oral use, which we have done extensively, or to even lavage of large wounds or abscess cavities. So I think that it's not a problem. That's my opinion.
All right, thank you very much.
Thank you.
So I will set a timer for you, and it will give you 1 ring at 2 minutes.
Okay. Well, first, thank you very, very much for the invitation being here today. And I'm going to change quite a bit topics here. I'm not going to talk about phage therapy almost at all. I'm more in the metagenomics and into the phage, trying to understand phage, which is kind of a challenging question in many different ways. So, okay, so why I like studying gut phages in particular, and I think I see it at very different levels. So I like, for one side, for example, the ecological importance, and there's very interesting community dynamics that lytic cycles offer. So as bacteria changes in the gut, also the phages will change with them. So they begin creating these dynamics in the community that's really interesting from the ecological perspective to study. There's also the evolutionary importance. So we have predator-prey dynamics that will happen with lytic phages. So this is going to be changing and adapting to different situations. So it'll be interesting also to see how those phages are changing over time. And then we also have the lysogenic cycles. So this is going to be very important for horizontal gene transfer and response to environmental signals. So what genes are phages moving in the environment, and what genes are phages giving into the environment. So it'll be nice to explore these too. And then the virome diversity, I believe this is a very important fingerprint that usually has better resolution than the bacterial level. So a lot of people are doing microbiomes now. They are looking at the microbiome profile. And I think bacteriophage, especially like in gut communities or any other body site associated community, it gives another layer of complexity into that fingerprint, which is very important. And on the other side, I like this, and probably you all agree with me that viruses or phages are essentially the best researchers. And this is why I usually think that phages are great researchers. First, they're 10 times more abundant than prokaryotes. This number keeps changing, but people believe that there are 10 times more phages than prokaryotes, so it's a very high number. They're present in all the environments when we can find a host, a potential host, so phages are everywhere. And their abundance in such a high number, 10 to the 30 is an estimate, makes them a masters of very large number game, making any probable event quite likely. Like, imagine winning the lottery. Winning the lottery is in the order of 10 to the, what, 10 to the 6, 10 to the 7? These are 10 to the 30. If they all play the lottery, they'll be winning it every single time with no chance of error. So they tried everything. And their mutation capacity and adaptation has allowed them to explore a lot of that adaptation landscape. However, they have limited carrying capacity. They cannot keep accumulating and accumulating DNA. They will only be carrying the DNA that will be useful for them. So in summary, they have done all the research. They have tried all possible options. They have looked for all possible genes, and they carry just the ones they need and the ones that they will be useful for surviving in a given host. But we understand very little of that genetic diversity. I think everybody that has to try to annotate a phage, and when looking at a phage genome, have seen that essentially everything is hypothetical. We just don't understand them. But they have in their genes the key to understanding very, very interesting biology. So that's why I think they're very interesting. So the initial characterization of human virome, well, my breadboard started some years before, but my first approximation to that was in 2010 when we did this set of studies. So it was 4 families, monozygotic, twin pairs and their mother, 3 different time points, 0, 2 months, and 12 months later. Purified VLPs, 454 sequencing at that time. And I'm only going to show this slide here is that the first thing we did was compare with a reference database at that time. And what we saw is that in average, 81% of the data didn't match anything in databases. So that was very surprising at that time, and we did some other analysis at that moment. And I'm not going to talk about much of those analyses, but what we saw at that moment was that most viral diversity was not previously described, but we postulated that the virome was dominated by temperate bacteriophages, or essentially in healthy adult humans in their gut, there was mostly prophages being induced at one time or another. It was mostly temperate bacteriophages. And some of that evidence, we think, it was we saw a high abundance of prophages and temperate phages by the little bit that we were able to annotate. There was a very low predicted number of virotypes per individual. Instead of the 10-to-1 ratio, we saw more like a 1-to-1 ratio. And there was a very high conservation in the rank abundance and sequence identity over time. So in those patients that we have 3 time points, when we look at them 2 months later, 1 year later, and we find the same phage, and when we compare the sequences, they were essentially the same. They were not changing at all, even over a 1-year period. And there was very high interpersonal variation. So when you compare the viral profile, meaning who's there and at what abundance, it was very different from one individual to another one. So this was back in 2010, and we keep looking a little bit on viruses. So another study we wanted to do was malnutrition to see what happened with viruses. And this was a collaborative project in Malawi following 317 twin pairs between 1 to 36 months, so very young twins. Why Malawi? Because it has 60% incidence of growth stunting, 2% twin rate. So that makes that 1/3 of the twins that we were studying at some point will develop malnutrition. And the rate of discordance for the twin pairs was over 50%. So the cohort included all these twin pairs at the beginning, so all were healthy. And over time, some of them remained healthy. We had some concordant for moderate or severe malnutrition, and some of them were discordant. When either one or both of the twins developed malnutrition, both were treated with therapeutic food, and samples were collected before, during, and after the treatment. So this was more or less the sampling scheme over the 36 months. We have different families, and we will try to collect at least 6 time points once there was an event of malnutrition during the therapy and after recovery of the therapy. So in total, we had 231 samples, average 56,000 sequences per sample. And the good thing was that we have a good sampling over time for those twins. But still, when we have this, and we compare to many different databases at that moment, and even cross against the databases, but the most common observation was that still most of the samples have like no hit to anything that is known. So this was in 2015, and still some of those samples, 99% of the reads, they will just not hit anything in any database. We're still completely unknown diversity. So we're like, okay, let's try to find some other way to try to study these viruses. And by that time, also, Vaz was working already in the cross assembly, so cross-assembly strategies. Let's take like the reads from the different samples and try to assemble those together. And since we have up to 6 samplings from the same individual, maybe if a virus was in one sample at some abundance and then in the next sample at a different abundance, that will help us assemble the genome. So we tried that. We got over 17,000 contigs. All the red ones were the ones that we have evidence that we, like one end mapped to the other end, so they are likely circular. So we have a lot of viruses around 6 kb, 7 kb, but we have large viruses, 100 kb, 200 kb, even 300 kb contigs. And with very high coverage. This is 100x average median coverage, 1,000x, 10,000x, almost 100,000x coverage. So some of those have really deep sequencing of those viruses. And the good thing here is that in average, we were able to use over 90% of the data. So now it's not that 90% of the data was not mapping, but now 90% of the data, we can actually use them to fall into this context. And since there are longer sequences, we can at least try to have a better annotation of those. So when we look at those viruses, well, still around 50% of those contexts have no similarity to databases. It was amazing to see a stretch of 200,000 base pairs that you compare to a database, and you don't have anything greater than 20 base pairs with similarity to those contexts. So really, really diverse, really different to what we know this far. But the other 50%, a good percentage was viruses, phages, a lot of those double-stranded DNA from the traditional families, but we have also a lot of eukaryotic viruses. And in this case, in particular, we have these anelloviridae viruses. So it is known that in kids, in particular, you know, the sampling was between month 1 and 36. So for these kids, we have seen a lot of anelloviruses, and it's quite common. It's now believed that around 90% of the population has anelloviruses colonizing at very young age. And then whenever there's immune suppression, you'll find those anelloviruses coming out again. So just the immune system keep them under control, but most of us has those viruses somewhere. In terms of diversity, it was very interesting to see that the same individual over time has relatively low diversity. Then when we were comparing twin-twin, they also have very close diversity. But remember that twin-twin here are very young kids. Kids living together, being fed together the same food. So they're sharing a lot of that early environmental exposure. Then the twin and the mother was very, very different. Even unrelated twins were almost as different as the twin and the mother. And between unrelated twin and a twin and a mother unrelated, that was the biggest difference. But then unrelated mothers were a little closer together. So it seems like adults have a little bit more similar viromes. Twins, even unrelated, they have more similar viromes. And when we compare unrelated twin and mothers, that's the most diverse you can get. So there seems to be an effect of early environmental exposure and even a little bit of age. So we tried clustering those profiles, and we identified a lot of these contexts. So each row here is a different context, and there is probably around 5,000 of those here. And this is each twin pair. Here we don't have the mothers. And you can see that over the 6 samplings of one twin and the 6 samplings of the other twin, there were a lot of those contexts that were very specific for that pair and essentially completely absent from everybody else. So it's still, as we saw with the previous study, that the viral profile seemed to be very highly individual. So in that case, it was adults. And in this case, since we have twin pairs and very young twin pairs, they still are sharing a lot of those viruses between the twin pair. So we saw this part. And we also did some other analysis, and we were able to see that even at this young age, if you take the viral profile, you will be able to find some contexts that correlate very well with age. So you can be able to classify the sample just by the viral profile and be able to tell with more or less a good accuracy what age was the twin that was the donor of that sample, just by the viral profile. And now, moving more recently, so one of my students, Laura, that is sitting back there, and she's going to have a poster on this study, so you can ask her later on about the study. So we're trying to expand that initial study. So that it's not anymore 36 samples. Now we have 22 families, 60 individuals, 140 samples. And one of the characteristics of those samples, we have lean, overweight, obese in the 3 different categories. But again, looking at the data right now, there's still no hits to almost anything. It's still the reads. We're still talking about 454 reads, but this is still not hitting almost anything in the databases. But we're still doing the assembly strategy. Again, we have around 17,000 contexts. We still have a lot of these contexts now, and now we know all these small, like 6, 7 kb contexts, they are microviruses. So that microviridae, and in particular alphaviridae, is a subfamily that is now described to be associated with the Bacterioidetes, and Bacterioidetes is a very important phyla in the gut. So most of these viruses are for that family. We don't have a lot of those of the Annelloviridae, because as I mentioned, Annelloviridae is more from the young kids. These are adults already, so we don't have a lot of those in here. And again, we were able to use a very high percentage of the data, over 90% of the data, almost 95% of the data, 96% of the data, into the assembly of these viruses. And something that was interesting here was that we were mapping a lot of those reads against a gut database. This gut database is gut-associated genomes. So it's a set of genomes that we know that are of bacteria that are commonly found in the gut. So we wanted to map those. And at the beginning, we saw, okay, this could be information about potential bacterial contamination of our sample. And if you see, some of these samples have large hits to that gut database. So, okay, we're like worried at some point, it's like maybe we do have some bacterial contamination, even though this was VLP purified, all of this, and these were all cesium gradient purified VLPs. We're like, maybe we have some bacterial contamination here. So what Laura did was have 100 kb windows and map against the genomes and look at those windows. Because one big problem that we have here is that it's really hard to tell apart when you have just by numbers of hits against a bacterial database, if you really have bacterial contamination or if you have prophages that are also present in those genomes. Because if you have prophages, you're going to have a lot of reads mapping to those prophages. So we wanted to see if it was contamination or not. So we were able to actually see that, yes, there was some contamination, mostly E. coli contamination. And this E. coli contamination also comes mainly from the reagents. So we have to do MDA amplification in some cases. And some of these reagents will have a little bit of DNA, E. coli DNA left over. So most of the contamination was coming from that. But we did have some Bacteroides, Akkermansia, and Ruminococcus contamination. So you can see coverage over essentially all the genome, but we didn't want to risk removing those prophages. So what we do is a normalized count. So we will see what is the average number of reads mapping through the genome, and we will see if there's any of those bins, around 100 KB bins, that differ from that average. So if it differs significantly, probably is that in that region you have a prophage, and you're having a lot of reads mapping to that prophage. So we will not clean those parts, but we will clean everything else. So that way we are removing bacterial contamination without actually removing prophages. And that is interesting because this also gives us a way of identifying prophages that we have experimental evidence, because we can recover the VLP information and map them to the genome. So now we're getting all NCVI bacterial genomes and mapping all the reads from the viroms there to potentially identify prophages from those genomes that have not been characterized previously. Again, we begin doing some tests, and what we have seen is that it's very highly individual. So here we still have 1, 2, or 3 samples per individual, and all these contexts, almost at least half of the contexts we were able to identify, they are really specific for one single individual. You will see that context in one individual and essentially nobody else. So there's still a large percent of the contexts that you will see only in a given individual and almost nobody else. Something else that we are just seeing, this is a very recent result, is that we even have some samples. If we sort all the samples by age, we will be able to find some samples that are present in more than one individual, even from different families. And the only thing that they have in common is that they are around the same age. Here is around 20, 30 years. Here are the mothers around 50, 60 years. So there are some viruses that seem to be more represented by age beings. So we haven't gone too deep into this. We haven't really checked what those contexts look like, but there seems to be something suggesting that there may be some context related to age too. But then Vaz will say, okay, here you're seeing a lot of individual contexts, but I've shown you, okay, I'll go back to that. But I've shown that there's one context that you can find in a lot of different metagenomes, and you find that context all over the place. Have you seen that context in here? It's like, yes, we have seen that context in here. In fact, we haven't seen it once. We have at least seen it 3 times among our different samples. So these 3 contexts, they all map to crossphage, and they're present in at least 90% of the individuals, not of the samples, of the individuals. So from those 60 individuals, we find crossphage in 3 of them. But interestingly, now we don't only find crossphage as being such an abundance phage, but we also have some other examples. So these 5 contexts, we were able to assemble those, and right now it's a context between those 5. It makes a 12 KB context of a phage we cannot really annotate more than this toxin-antitoxin protein pair, but it's abundant in a lot of those samples too. Some of those have also crossphage, some of those don't have crossphage. And then there's this other context, that's an 88 KB context. It's very long too. It's also present in a lot of the individuals. So the profiles are very individual, but that doesn't mean that there's no viruses that are very ubiquitous, and we'll find those viruses in many of the individuals. This slide that I skipped, we also tested if there was context associated with the BMI. We don't have very highly significant results, but there are some contexts that are enriched. So there are some contexts here that are enriched in the lean, in the overweight, in the obese category 2, and in the obese category 3. So there are some contexts that seem to be related with at least enriched in different levels of obesity, and we're still trying to characterize and annotate some of those. So going back to the crossphage, it's not only a couple of contexts. We have 49 contexts that look like crossphage. Even 5 of those look like full contexts. And when we align those with crossphage, it has very nice alignment, and some regions, high diversity regions along the genome. So it seems that crossphage is really abundant, and we have different versions of crossphage coming from different samples. And probably these regions of divergence probably could be mediating a little bit of host specificity or something like that. So we are still looking into this. But looking at this data, what we can see is that we need more tools to analyze those viral metagenomes. There's still a lot of those contexts, a lot of those reads that we just don't know what they are. A lot of the people that were in the workshop this morning, they know that annotations tool depends completely on what is outside in the database. And right now, that is very, very limited. So we need better metagenomic assembly, ways to identify viral family markers, and ways to extract information from unknown common viral contexts. Now we're finding these contexts that we find them in a lot of different individuals, but we still don't know what they are. We still don't know what they code for. So we wanted to extract a little bit of that information. So another master's student in my lab, what he did was trying to get more information from viruses. So he downloaded every single viral genome in NCBI, 44,000 genomes. After dereplicating, just removing things that were essentially exactly the same, that number reduced to almost 14,000. So that's as many viruses as we know right now. That's all. From these 14,000 viruses, 442,000 proteins. And then, since two different proteins may have the same domain and they're not necessarily shared by evolution, what we did was fragmented into protein segments or domains using PFAM, and we were able to get 693,000 of those protein segments. Then we used just a software to generate orthologs. So how many of these protein domains or protein segments are shared between the different viruses? And with that, we were able to identify 21,000 of those orthologs. We're calling those BFOGs right now for viral and phages ortholog groups because it's including everybody there. And these 31,000 BFOGs are shared between those 14,000 that inherently are those 44,000 genomes. So this will create a matrix of all the genomes and all those 31,000 BFOGs and their abundance. So this could be used as an option for classification. So now we can use these and machine learning algorithms to see how well these BFOGs are capable of classifying viruses. So we test that at the order level, family level, genus level. These are the number of genomes we have. This is the number of classes. So there are 7 different viral orders, 83 different viral families, 335 different genuses. And this was the level of classification we were getting. So we can just take one virus, and depending on the BFOGs they had, 96.9% of the time, we were able to classify the right genus, 98.3% of the time, the right family, and 99% of the time, the right order. So these are highly discriminatory. So here's the confusion matrices for some of those classifications, but then we were able to find those BFOGs. So we have, for each of the different viral orders, some of those signature BFOGs. So we know their abundance for viral order, or even the 98 viral families that we are now testing. So there's a lot of these contexts, and it's not a very large data set of those protein domains that are completely conserved, and there are signature of each of those viral families or viral orders. So you can use those to classify new viruses. And the other thing you can use is that use this information to create a dendrogram out of all viruses. So we created this, and you can see it makes a very nice dendrogram where all phages, like all Cadoverales, are together. Right next to it, herpes viruses that may have a few over here. And we have all different known orders, usually like completely monophyletically classified. Interestingly, all the ones in yellow, they don't have any described order, but you can see consistent clustering of those two. So this could be even used to propose like new orders or new taxonomical levels that can be used based on the sequence of those viruses. And then we can also use these domains or conserved domains in order to begin looking for new version of those viruses out of metagenomic data sets. So what we also did in collaboration with Arthur Gruber in Brazil was to try to make a software that we take those domains, conserved domains of proteins, and we were looking at some of this morning, and just this is BP1 protein of the alpha-virine, and just the alpha-virine, and you will make an alignment, select a given region that is more or less conserved. You see it's not highly conserved, but you can generate a model, a statistical model of that variation, and use that as a seed to generate an assembly of those viruses. And with this, we were able to assemble a lot of the viruses from this family, so from a set of only 36 samples, we were able to assemble 45 contigs. And those microviridie, you know, those are between 6 and 7 kb. So at least 20 of those genomes that we assembled were complete genomes. And when we do a phylogenetic tree based on the BP1 reconstruction, we can see they're all exclusively alpha-virinate. They don't hit any of the other families. So really, there are very specific markers that you can get that you can use to identify novel viruses from a given family. So we are using now these tools in order, like, we pick a family and we can go to metagenomic data sets to see how much more we can assemble from that family of viruses, expanding what we can find of reference viruses for annotation. So in summary, most viral diversity is still very unknown. There are viral sequences and viral families that are conserved, and we'll find them especially in different environments. And some of the new tools that we're trying to make begin to characterize or exploit those unknowns. And that will help us probably, hopefully, to explore a little bit of that biology dark matter that is still all that information that is coded in viruses. And with this, I just want to thank, in particular, my group in Colombia, the work of my two students that I was showing today, Laura and Leonardo. I also want to invite you guys to see Ruth Posters. She's a PhD student that is starting her work in phages. And of course, my collaborators during these years in a lot of this work. And thank you guys for being here.
All right, we have some time for a couple of questions, and then we'll move on to the next stop. Questions, please.
Forgive the very general question, but how is nanopore sequencing influencing your research at this moment for lentiviral analysis?
I haven't tried to look at nanopore sequencing yet. I'm really curious about it. I think it's very promising in the fact that it's going to generate very long reads. It's still the error rate. I was chatting with the people from Nanopore a few months ago. It's still, I think, is quite high. But I think it's very useful. I've seen a lot of studies in field where they can go, like with the CCAP and the epidemic. That was recently. They were in Brazil in different places sequencing the virus. And I think it will be a very nice source of sequence variation, although the error rate kind of limits a little bit of that. But for viral discovery, I think it could be useful. I haven't used a lot of that data yet. I haven't really checked any of that data yet.
Hi, Alejandro. It's a very interesting talk. I have questions about your, so the bacterial sequences that get mapped in your data. So would it be easier if you map, like if you try to filter by mapping them to core E. coli genome, for example, to get rid of most of the prophages, or I mean, does it help, or you still get?
To get rid of like the contamination, so
to figure out if it's contamination or just mapping to prophages.
Right. So I think the tricky part is not only, so you have the prophages that are long segments that can get induced or not, but then you have pseudophages too, and then there are the genes that are being mobilized by phages. So sometimes integrases, they could be in some place and it'll be just that little gene, but that was also present in a phage. So all that part makes it very tricky. It's not like black and white. It's not like this prophage and nothing else, but there are different regions. So at least what we tried is to reduce that by binning, like 100 kb bins, usually kind of guarantee that hopefully the phage will fall within 1 bin or maximum 2 bins. And that way we narrow down the window, and then we look at that window, and if we see continuous mapping over a stretch that look like a prophage, then we'll leave that intact and remove like everything that is else that is around.
And I have a question. I don't know actually. Does the gut microbiome have any GTAs like in marine bacteria? Like, do you have gene transfer agents, or this hasn't been shown in?
That's a good question, and sometimes because of this precisely, we've been discussing a lot about it, because it's kind of,
I know the people that did a lot of these extractions. I myself did some of them, and I know the quality of those VLP extractions. It was cesium grading, purification, and everything. So, and it's not random. The type of genomes that we're finding almost consistently the same genomes when they were present. And it's not because you'll expect, like, if the genome was in high abundance, then you'll have higher chances of seeing that genome as a contamination, and that's not really the case. So it's probably likely that those bacteria are probably generating some sort of vesicles or something where they're packaging some of that DNA that get co-purified, or maybe even GTA or something like that. We still don't know what may be happening, but I don't think it's as simple as just bacterial contamination. There may be something else in there.
All right, one more question, please.
Yeah, this is just related to Rami's question, but could it be that these sequences are ending up within these phages somehow? And could you test that maybe by checking paired reads, or where?
Yes, that'll be something to check. In these studies, we don't have parent reads. This is still all 454 single end, but that could be an option. In previous cases, we have tried to look for something like that, and we haven't seen any observation of like a read that half maps the phage and half maps the other part, like the excision of the prophage is cut in the wrong places or something like that. We haven't seen any evidence of that, but I haven't looked at that in high detail, so we can check on that.
All right, thank you, Alejandro.